Mutations. Mutations are permanent changes in the genetic material of organisms. Any kind of mix-up of nucleotides in a genome is a mutation. Mutations that concern us are ones that occur to genes. Nucleotide problems in the non-coding regions of the genome is not very significant to the developmental and reproductive capacity of the individual. So mutations then refer to changes in the genetic material at gene positions. Mutations occurring to any of the somatic cells can cause problems to that particular individual, but are not transferred from one generation to another. A mutation in the somatic cells of a developing fetus is hugely problematic because while those cells are continuing to divide, the mutation is replicating right along with them. Women of childbearing age are always asked if they think they might be pregnant before undergoing medical procedures that involve mutagenic agents like X-rays. Somatic cell mutations are a key cause of cancer. We'll discuss the hereditary links to cancer later. Mutations in the gametes, germline mutations, of an organism can pass the trait down to future generations. Mutations occurring in the gametes risk affecting the offspring. A sperm cell carrying a mutation that fertilizes a normal egg transfers that mutation to every cell of the offspring. Point mutations involve one or two nucleotides at a single point in the gene. Another way to visualize how a point mutation and the addition of a single nucleotide ruins the formation of a protein is to consider this sentence. The big red fox ate the egg, purposely done in three-letter words to represent DNA codons. Each would transcribe into mRNA codons and translate into amino acids. Add in a nucleotide represented by the X, and the rest of the codon sequence is gibberish. A point mutation where a nucleotide is deleted yields a similar problem. Deleting the nucleotide represented by the R in red causes the rest of the sequence to jumble. Frameshift mutations caused by addition and deletion point mutations. Point mutations like this are called frameshift mutations because the mutation shifts the frame in which the nucleotides are read as codons. Frameshift mutations are mostly always nonsense mutations. Nonsense mutations mean the genes cannot produce a functional protein. Another type of point mutation is a substitution mutation. Here's part of a template strand of DNA. After transcription to mRNA, the amino acid sequence following translation is valine, histidine, leucine, threonine, proline, glutamate, and another glutamate. This sequence of amino acids make up part of the beta-globulin polypeptide, one of the two polypeptides that combine to form the protein hemoglobin, the oxygen-carrying pigment of human red blood cells. The thymine nucleotide is highlighted here to represent the location of a mutation from thymine to adenine, which changes the glutamate amino acid to valine. The substitution of an adenine in place of a thymine changes a single amino acid and the structure and function of the resulting protein. In this case, there is a significant drop in the protein's ability to carry oxygen, and due to the millions of hemoglobin molecules in a single red blood cell taking on a deformed shape, that deformity affects the overall shape of the red blood cell, which takes on a crescent moon or sickle shape. This is the mutation that causes sickle cell anemia. Substitution mutations can be missense mutations. It can also be a nonsense mutation when the protein product is useless. For example, the altered codon might code for a stop codon and interrupt protein formation altogether. But in the case of sickle cell anemia, the protein has a reduced capacity, so we call these missense mutations. Missense mutations are not necessarily bad, usually they are, but the protein produced might actually improve its function and be responsible for the evolution of species. If a substitution mutation caused the adenine here to change to a guanine, The resulting amino acid would change from a threonine to a, well, still threonine. This is called a silent mutation. The mutation does not cause a change in the amino acid sequence. These are all examples of point mutations. Mutations covering a larger amount of DNA are called chromosomal mutations and can affect multiple genes. These kind of mutations can result in the loss or duplication of portions of chromosomes. We'll talk more about these kinds of mutations and their link to cancer later. Spontaneous mutations occur due to molecular interactions within the cell, usually caused by enzymes pairing incorrect bases during DNA replication. Induced mutations are due to exposure to mutagens, of which there are two general categories. Physical mutations physically changes the structure of DNA. Any mutagenic agent that can physically be applied changes to random portions in DNA is a physical mutation. For examples, X-ray and UV radiation. High-energy radiation, such as these types of radiation, cause DNA to fracture, resulting in point mutations. DNA can even tear apart, causing imprecise repair as enzymes apply fragments to different areas of the chromosome or a different chromosome altogether. The body's defenses against UV radiation, for example, include the production of melanin by the skin cells. Melanin limits the penetrative abilities of UV radiation, absorbing most of it before it reaches the nucleus where it can damage the DNA. People who are fair-skinned produce insufficient amounts of melanin for what was once regarded as normal exposure to the sun. The application of sunblock creams assists in absorbing the UV radiation before it can penetrate the cells. Chemical mutagens are molecules that enter the nucleus to induce mutations, for example, free radicals and nitrites. Free radicals are molecules that have an unpaired electron. Free radicals damage cells by oxidizing the cell's molecules, and evidence supports the notion that free radicals in the environment are responsible for our aging. The consumption of chemicals that more readily react with free radicals before they have a chance to react with our bodies is a big business in the world of nutritional supplements. Antioxidants work to neutralize the effect of free radicals before they can damage cells or cause mutations in the DNA. Nitrites are found in some food preservatives and can act by affecting the sequencing of nucleotides during DNA replication. Other chemical mutagens include gasoline fumes and a whole rat's nest of chemicals found in cigarette smoke. Mutations in cancer. In this model of a piece of chromatin, the structural gene is an oncogene, a gene that codes for a protein that directly influences cell division. Understandably, a gene like this needs to be regulated. Uncontrolled expression of this gene is one of the contributors to cancer. If a mutagenic agent like X-rays breaks the DNA apart and then glues them back together, and the regulator gene is no longer in a position to control the expression of the oncogene, then the oncogene is free to produce proteins that trigger cell division. It's important to point out that no one mutation causes cancer. It is understood that cancer can be caused by about 6 mutations, and the mutations have to be specific and affect only the genes responsible for cell division and tumor suppression. This is why generally it's the elderly who get cancer. They have had a lifetime of acquiring enough mutations so that at least 6 of them will have affected change on the key genes. What about those people who get cancer at a young age? Well, the most likely reason is that these people have, due to heredity, an overabundance of oncogenes or a lack of tumor suppressor genes, or both. This heredity aspect explains why some people who smoke might avoid cancer altogether, while other smokers may suffer cancer earlier. Also, some viruses can cause cancer, either by injecting the host cell with genetic material that damages the host genetic material, or by even injecting the host with more oncogenes or genes that cause the overexpression of the host's oncogenes. For instance, the relationship between cervical cancer and the human papillomavirus is so well established that women who have never had sex don't require pap tests. So mutations in cancer. Mutated genes that are linked to cancer are oncogenes and tumor suppressor genes. At least 6 specific mutations are linked to cancer. Other contributors: gene amplification. The overexpression of oncogenes is also known to be a risk factor for developing cancer. The underexpression of tumor suppressor genes is also a contributor. A person with this inherited trait and an engager in high-risk lifestyles like smoking, being fair-skinned and suntanning, etc., increases the likelihood of developing cancer.